and CEO, Daphne Zohar, our co-founder and board chair, Dr. Steve Paul, our co-founder and CSO, Dr. Michael Chen, and our CFO, Lauren White. Members of our team not on today's call include our Chief Medical Officer and President of Clinical Development, Dr. Tony LaBelle, and our General Counsel, Lana Gladstein. With that, I will turn the call over to Daphne.
Thanks, Adam. Welcome to everyone who's joining us for this roadshow presentation. Seaport is a neuropsychiatry company with a clinically validated platform that has already generated a robust pipeline of 3 programs, 2 of which have now shown initial clinical proof of concept. Our most advanced program is in a phase 2b study, which has the potential to be registration enabling. Our second program recently achieved a key phase 1 proof of concept milestone, and our third candidate is in preclinical development. All of our programs are built on mechanisms that have already shown human efficacy data but were previously held back by limitations glyph can now address. Glyph can overcome issues like poor oral bioavailability, high PK variability, and related side effects. And every time we glyph a molecule, we generate a new chemical entity with novel composition of matter IP. We have a seasoned leadership team with a proven track record of bringing new psychiatric medicines to patients and driving successful business outcomes. And we have ample capital to carry us through the next key clinical milestones for each of our lead programs. There are over 1 billion people globally living with mental illness, which is comparable in prevalence to obesity or hypertension. At Seaport, we're focused on depression and anxiety, each of which affects over 300 million people globally. Depression and anxiety are often comorbid. Approximately 60% of patients with major depressive disorder, or MDD, also have anxious distress, and roughly 60% of patients with generalized anxiety disorder also have MDD. And these conditions are not only disabling, they can also be deadly. One statistic I find particularly disturbing is that suicide is now a top 3 cause of death in adolescents. There's a clear need for better medicines. Existing psychiatric medicines have helped millions of people. Our co-founder, Steve Paul, who you'll hear from shortly, helped develop Zyprexa and Cymbalta, the top 2 medicines on the list on the left. These medicines are among the most widely prescribed and commercially successful drugs of all time. With that said, they have limitations. They have modest efficacy. A third of patients don't respond, and approximately half of patients are unable to achieve remission with first-line therapies. These medicines take weeks to work, and they have side effects like sexual dysfunction, weight gain, and sleep disturbance. Seaport is positioned to outperform across all 3 of these parameters. And this is a great time to be working on these big problems in neuropsychiatry, with a neuroscience renaissance taking place, both on the part of the science and also renewed interest from pharma companies as they look for the next obesity-like opportunity to make an impact on society. With this excitement and tremendous unmet need, we need to acknowledge that there are challenges that have held back this field. The first is that unvalidated mechanisms have had high failure rates in the translation from preclinical to human studies. That's why we're starting with mechanisms that already have proven human efficacy, but were held back by poor drug-like properties and related side effects. These are properties we can apply glyph to overcome. The final point is that running clinical studies in neuropsychiatry has been challenging. It's important to have a team that has experience in designing and executing studies to manage placebo response, ensure qualified patient participants, and select the right sites. Our team has a lot of experience with this, and we're applying many of the same approaches that worked well for Karuna, which ran 3 successful registration-enabling studies. The approach we're taking at Seaport has other similarities to what we did at Karuna. At Karuna, we knew that xanomeline was an effective antipsychotic from data generated under Steve's leadership at Lilly. But it was sitting on a shelf due to GI tolerability issues that we were able to address with the invention of KarXT, now called Cobenfy, at my former company, PureTech. At Seaport, we're using the same approach of identifying drugs with proven efficacy, addressing the limitations that have held those drugs back, this time with glyph, and then designing and executing studies with an experienced team. In other ways, we have advantages that we didn't have at Karuna, including the glyph platform, a robust pipeline, and novel composition of matter IP, including issued composition of matter IP on our lead program. Glyph was initially invented at Monash University by world-leading experts in lymphatic transport, Dr. Chris Porter and Dr. Jamie Simpson. Jamie is now head of chemistry at Seaport. We brought the Glyph technology into PureTech and worked on it for 5 years before Seaport was spun out. During that time, we glyphed 24 molecules and achieved initial clinical proof of concept for the platform and our lead program. Let me tell you how it works. Glyph is a prodrug technology which cloaks a drug so that the body recognizes it as if it were a dietary fat and traffics it through the lymphatic system, bypassing the liver and transporting it via a side road directly into the bloodstream, as you can see on this slide. Because less of the drug is metabolized by the liver, more drug can get to the brain. By bypassing the liver, Glyph can overcome issues such as liver toxicity, low oral bioavailability, variable PK, and it enables us to reach therapeutic exposures at a lower dose. It also enables drugs that otherwise need to be given intravenously to be dosed orally. We've applied Glyph to generate a pipeline of novel treatments for depression, anxiety, and other neuropsychiatric disorders. On this slide, you can see the key milestone timing on the right. Glyph Allo, which is being advanced in a phase 2b study for major depressive disorder, or MDD, has top-line data expected in the first half of next year. A top-tier journal, Science Translational Medicine, recently published a peer-reviewed paper detailing the progression of this program from discovery to clinical proof of concept. The paper is posted on our website. Glyph Ego is being advanced for generalized anxiety disorder, or GAD. Following the positive phase 1 proof of concept data that we announced earlier this month, we plan to initiate phase 2A and phase 2B trials, both of which we expect to read out in 2028. Our third program, Glyph 2B-LSD, is in preclinical development with first-in-human enabling studies to be completed by end of next year. We retain exclusive worldwide development and commercialization rights to all of our programs. Glyph has broad applicability beyond CNS, and we can help other pharmaceutical companies and generate non-dilutive funding through partnerships while we remain focused internally on neuroscience. We can also advance non-CNS programs with grant funding. For example, we recently announced a grant of up to $15 million from ARPA-H for a glyph molecule specifically engineered to target and normalize dysfunction in the gut lymphatic system, which plays a central role in metabolic disease, pancreatic cancer, and immunity. I'll now turn it over to Steve to provide an overview of our Glyph Allo program before we dive into the data. Steve?
Thanks, Daphne. Glyph Allo is based on an approved drug. It's an oral prodrug of allopregnanolone, which is an endogenous molecule that has been clinically validated in third-party trials as a rapidly acting antidepressant with anxiolytic, anti-anxiety, and sleep-promoting effects. Despite its efficacy, the scope of allopregnanolone's clinical use has been constrained by its very low oral bioavailability. In fact, without Glyph, over 95% of allopregnanolone doesn't make it to the bloodstream when administered orally. While allopregnanolone has been approved for the treatment of a form of depression known as postpartum depression, it was only available as a 60-hour IV infusion. A synthetic analog of allopregnanolone called zuranolone was also advanced by Sage for MDD, major depressive disorder, and in fact met its primary endpoint in 5 out of 6 studies, and was approved recently in Japan for MDD, but was not approved by the FDA. We believe that there were some clinical trial design and execution factors that played a key role in preventing it from being approved for MDD here in the U.S. Let's talk about those clinical design features. Zuranolone showed strong and consistent efficacy in MDD trials, meeting the primary endpoint in 5 of 6 of those trials. Importantly, it also showed a consistent rapid effect, but FDA guidance requires that a drug show efficacy at 6 weeks. Zuranolone was only dosed for 2 weeks, showing a deepening of response over that 2-week period, but did not show efficacy at 6 weeks when patients had been off drug for a month. We plan to dose for the full 6 weeks with Glyph Allo. One other learning we plan to apply in our studies is to limit the number of clinician-administered assessments. Our team at Seaport did a meta-analysis of MDD trials over the last 10 years, and one takeaway was that the frequency of clinician assessments is significantly associated with a larger placebo response. This makes sense because those interactions, like psychotherapy, may have had an effect. If you look at the figure on this slide, the 3 purple circles on the right are SAGE studies with zuranolone. They had double the number of clinician assessments than is typical. As you can see, they also saw a placebo response that was significantly higher than what is typical. So with GlyphAllo, we are optimizing the number of clinician-administered assessments. We will also be doing a number of things that we did at Karuna, including limiting the number of active arms in the study and scrutinizing patients and site selection, which Michael will describe in more detail. Michael, do you want to walk us through the data we've generated to date on GlyphAllo and the design of the BUOY-1 trial?
Sure. Thanks, Steve. In preclinical studies conducted in non-human primates, we've shown that GlyphAllo provides a greater than 9-fold increase in bioavailability compared to unmodified allopregnanolone. In the clinic, we've shown that once daily oral dosing of GlyphAllo provides similar exposures as those achieved with a 60-hour intravenous infusion of allopregnanolone. Now, in a separate third-party clinical trial, the oral bioavailability of unmodified allopregnanolone was shown to be very low, since 95% approximately of unmodified allopregnanolone is chewed up by the liver. Now, as Daphne mentioned, we recently published a paper in Science Translational Medicine detailing the robust preclinical and clinical data package we've generated with GlyphAllo to date. This includes data showing that peak allopregnanolone plasma levels and pharmacodynamic markers, including beta EEG power, saccadic eye velocity, and self-reported somnolence, were reached 3 to 5 hours post-dose. These data also show that we have therapeutically relevant exposures of drug circulating in the body and have dose-dependent pharmacodynamic effects in the brain consistent with allopregnanolone. And these return to baseline 6 to 8 hours post-dose. And turning to safety, GlyphAllo was generally well tolerated and in line with its pharmacology as a GABA-positive allosteric modulator. The most common reported AE was somnolence, which was mild and transient. There were also no safety signals attributed to Glyph itself, and we've now observed this in each of our 2 lead programs. Based on these data, we progressed GlyphAllo into a placebo-controlled phase 2A trial, where we showed that GlyphAllo potently blunted the acute physiological stress response in a validated clinical model of anxiety called the Trier Social Stress Test, or TSST. In the TSST, healthy volunteers are put in the rather stressful situation of having to prepare and give a speech, and then do live math in front of live judges. And as you can imagine, this causes levels of cortisol, the body's primary stress hormone, to spike. And this can be measured via a simple cheek swab. In the figure on the right, the gray line is the placebo group and the blue line is GlyphAllo. As you can see, oral administration of a single well-tolerated dose of GlyphAllo, 375 milligrams, achieved a statistically significant reduction versus placebo in the increase from baseline to peak levels of salivary cortisol, the p-value of 0.0001. The only neuropsychiatric drug to demonstrate a similar result on the TSST is alprazolam, a benzodiazepine that has been marketed as Xanax. So what we have here is a potential novel, rapidly acting treatment for major depression, which may also have benefits for anxious distress and sleep, and has the potential to avoid side effects like sexual dysfunction, weight gain, and sleep disturbances that can be caused by current treatments. Building on the supportive data that we've generated in phase 1 and phase 2A trials of GlyphAllo, last summer we initiated the phase 2B BUOY-1 trial of GlyphAllo in adults with MDD with or without anxious distress, which we believe has the potential to be registration enabling. This trial is currently enrolling and is expected to include up to a maximum of approximately 360 patients, randomized 1-to-1 to receive either GlyphAllo or placebo once daily at bedtime for 6 weeks. The primary endpoint of the trial is the change from baseline at 6 weeks in this Hamilton Depression Rating Scale, or HAM-D, a regulatory standard depression severity rating scale. Applying our team's deep expertise in neuropsychiatry clinical development, we've implemented a number of key design elements aimed at improving the probability of success in this trial. As Steve mentioned, we are treating patients for the full 6-week duration of the primary endpoint timeframe, as opposed to previous trials that treated patients for only the first 2 weeks. We're also enriching for patients that have MDD with anxious distress. To reduce the placebo response, we are optimizing the frequency of clinician-administered assessments and including a single treatment arm, as both the frequency of assessments and the number of active arms in a trial are known to increase the placebo response. And we are scrutinizing site selection and patient enrollment to ensure appropriate diagnosis and to exclude inappropriate patients. To optimize the sample size, we've included a pre-specified sample size re-estimation, or SSRE, to provide the potential to re-estimate the total sample size at an interim point of the trial. This is not a futility analysis or an early efficacy analysis and is a fairly routine practice that may modestly pull in timelines. The sample size may be reduced following the SSRE, but will not be increased beyond the 360 patient number. I would also note that our expected timing of top-line data in the first half of next year is based on that full sample size of up to approximately 360 patients. And finally, while all somnolence observed in the phase 1 trial of GlyphAllo was mild and transient, we've worked with FDA to address some of their questions around excessive somnolence seen in a handful of patients on IV intravenous brexanolone. To that end, we've implemented a couple of design elements into BUI 1 to provide further reassurance around the differentiation of GlyphAllo in that regard. First, we conducted intensive safety monitoring on approximately 40 patients enrolled in the U.S. with oversight by a data monitoring committee. In these patients, no events of loss of consciousness, syncope, excessive sedation, falls, or hypoxia were reported, and intensive safety monitoring in BUI 1 has been discontinued. And second, patients enrolled in the U.S. are not driving during the study. Now, these elements are unique to patients in the United States. For patients enrolled in the EU, there is no intensive safety monitoring and a shorter 6-hour driving restriction. And as part of the broader GlyphAllo development program, we also plan to conduct additional studies, including a driving simulation study. We plan to initiate this driving simulation study of GlyphAllo by the end of the year, with data from this study ahead of the expected top-line readout of BUI 1. Let me now turn back to Steve, who will describe our second program, GlyphAggo.
Thanks, Michael. Like GlyphAllo, GlyphAggo is also based on an approved drug. GlyphAggo is a novel candidate for generalized anxiety disorder, or GAD, for which we recently announced positive top-line data from a phase 1 proof of concept trial. There's a significant unmet need with over 300 million people with anxiety disorders globally and 100 million people living with GAD. There have been no new therapies in almost 2 decades, the last one being Cymbalta, a drug I helped develop at Lilly, which was initially approved for depression. Agomelatine acts via a novel mechanism. It's a melatonin receptor agonist and a serotonin receptor 2C antagonist. And put simply, agomelatine works in generalized anxiety disorder. Agomelatine has been evaluated in 4 randomized placebo-controlled studies in GAD, and agomelatine statistically separated from placebo in all 4 studies. Needless to say, that's a pretty good batting average in neuropsychiatry. But it has one issue: it's a bit rough on the liver. Due to very high first-pass metabolism, over 90% of agomelatine is lost to the liver. This can cause high PK variability and liver enzyme elevations, which requires that patients treated with agomelatine undergo frequent liver function tests before starting treatment, after 3, 6, 12, and 24 weeks, and when increasing the dose. As a psychiatrist, I can tell you psychiatrists are not keen on doing frequent liver function testing. This burdensome liver testing, combined with the lack of patent protection, has limited agomelatine's adoption. GlyphAgo is designed to overcome this limitation by avoiding first-pass liver metabolism and achieving therapeutic exposures of agomelatine while reducing PK variability and reducing or eliminating the need for liver monitoring. I'd also like to point out that we believe this program has some important similarities to the KarXT program at Karuna. In the case of KarXT, we knew that xanomeline was an effective antipsychotic from the data we generated on it while I was at Lilly. The issue with xanomeline was its tolerability profile. Since it also stimulated muscarinic receptors in the periphery, it caused cholinergic adverse events like nausea, vomiting, and diarrhea. So the key question for the KarXT program wasn't whether xanomeline worked, but rather could the addition of trospium improve that tolerability profile? And this was a question we were able to address in phase 1. In the case of GlyphAgo here at Seaport, we know that agomelatine is effective in GAD, but its uptake has been limited by those liver enzyme elevations I mentioned. So the key question here is whether GlyphAgo can provide therapeutic exposures of agomelatine at a lower dose that will avoid those liver issues. And similar to KarXT, we believe we have addressed that question in phase 1, and Michael will walk you through the data shortly. As a review of the data on agomelatine in GAD, on the right on this slide is a figure from one of the 4 third-party randomized controlled trials in GAD I just mentioned, where agomelatine statistically separates nicely from placebo. Agomelatine's efficacy and tolerability in GAD is actually better than standard of care drugs like benzodiazepines, which have abuse liability, and SSRIs, which can cause sexual dysfunction, weight gain, and insomnia. Agomelatine also improves sleep latency, sleep architecture, and daytime sleepiness compared to SSRIs, which can worsen sleep and cause insomnia. This makes agomelatine well suited for GAD, where sleep disturbance is a core symptom. Michael will now touch on the liver issues agomelatine can cause in a bit more detail, and then review our phase 1 top-line data.
Thanks, Steve. The frequency of liver enzyme elevations with agomelatine is dose-dependent, as you can see on the figure on the right side of this slide. These liver enzyme elevations reach a maximum of 2 to 5-fold greater than placebo and are dependent on the amount of drug actually dosed. So as Steve mentioned, the goal with GlyphAgo is to bypass first-pass liver metabolism and deliver therapeutic AGO exposures, but lower the dose. If we lower the dose, that means less drug hitting the liver, which means less liver exposure, less liver enzyme increases, and the potential to reduce or eliminate liver function testing. And this requires increasing the bioavailability of agomelatine. Based on our preclinical studies of GlyphAgo in non-human primates, as well as computational modeling of liver enzyme elevations using DILIsym, as shown on this slide, we project that increasing the bioavailability of agomelatine by 2-fold with GlyphAgo will reduce or eliminate liver enzyme elevations and accordingly reduce or eliminate the need for liver function testing with agomelatine. As noted, we recently announced data from a phase 1 proof of concept trial aimed at determining whether we could meet this 2x bioavailability bar. Diving into the data, we were very pleased to see that in this trial, GlyphAgo demonstrated a 6.8-fold increase in bioavailability compared to unmodified agomelatine, significantly exceeding that 2-fold bar. The 6.8-fold increase in bioavailability is based on the head-to-head crossover portion of this phase 1 trial, shown in the figure on the right side of this slide. In this part of the trial, patients received GlyphAgo, and after a washout period, then received unmodified agomelatine at the approved 25 milligram dose. As we've discussed previously, the high first-pass metabolism of agomelatine in the liver can also result in high PK variability, which we also observed here, with multiple participants having low exposures of agomelatine despite dosing at the approved 25 milligram dose. In contrast, we found that glyphago reduced this PK variability by a factor of 10, resulting in more consistent exposures across participants. And consistent with this improved bioavailability, we also found that glyphago achieved a 1.9-fold increase in dose-normalized Cmax compared to unmodified agomelatine. In a separate portion of the study, shown here, healthy volunteers received a single 25 milligram dose of agomelatine or single ascending doses of glyphago. Here we demonstrated that glyphago had a dose-dependent 9.6 to 14.5-fold increase in dose-normalized exposure, as you can see on the figure on the right side of this slide. The relative increase in bioavailability was greater in the single ascending dose portion, since the crossover portion included participants who were taking estrogen-containing oral contraceptives, which are known to increase agomelatine exposure due to a drug-drug interaction in the liver. In the crossover portion, we saw no effect of oral contraceptive on glyphago exposures, further supporting glyphago's ability to bypass liver first-pass metabolism. Turning to safety, across all evaluated dose levels, glyphago was well tolerated, with no serious or severe adverse events observed and no clinically significant changes in liver-related laboratory parameters, including ALT, AST, or bilirubin. All treatment groups, including glyphago, unmodified agomelatine, and placebo, were well tolerated, and no serious or severe adverse events were observed in the trial. Taken together, we believe these results show that glyphago delivered the same exposures that are known to be efficacious with agomelatine, but with a lower dose, which means less agomelatine hitting the liver and with a more consistent PK profile. With these data in hand, we plan to kick off 2 additional trials of glyphago in the near term. The first of these is a phase 2A proof of pharmacology trial designed to evaluate the potential sleep benefit of glyphago in patients with GAD and sleep disturbance. As a reminder, agomelatine has been shown to improve sleep quality compared to an SSRI in patients with major depression, but this has never been evaluated in generalized anxiety disorder. Given that sleep disturbance is a core symptom of GAD, we believe glyphago may provide a differentiated benefit on sleep in GAD, and we expect top-line data from this phase 2A trial in early 2028. In parallel, we plan to initiate a randomized, double-blind, placebo-controlled phase 2B trial designed to evaluate the efficacy and safety of glyphago in patients with GAD. We believe this trial has the potential to be registration-enabling, and we expect top-line data from that trial by year-end 2028. I'll now hand it back over to Daphne to touch on our third program before we wrap up with a summary of our milestones and financials. Daphne?
Thanks, Michael. Our third program, GlypH2B-LSD, is a non-hallucinogenic neuroplastogen. In other words, it harnesses the pharmacology of a psychedelic, but without the trip, without the hallucination. This program builds on the growing body of evidence that psychedelics have the potential to achieve rapid, long-lasting treatment effects in psychiatric disorders. Despite the excitement around psychedelics, significant challenges remain, including functional unblinding of clinical trials due to hallucinations, the need for supervised administration, and concerns over long-term safety, such as the risk of fibrosis. We view compounds that can harness the pharmacology of psychedelics, but avoid the trip, as a large potential opportunity. And with GlypH2B-LSD, we aim to do that. GlypH2B-LSD is based on an analog of LSD that has been dosed in humans, shows activity in a rare headache disorder called cluster headache, and importantly, does not cause a trip. Preclinically, it shows 5-HT2A agonism, induces neuroplasticity, and looks like an effective antidepressant in animal models. This program is currently advancing in first-in-human enabling studies, which we expect to complete by the end of next year. And with that, I'll hand it over to Lauren to walk you through our milestones, runway, and expected use of proceeds.
Great, thanks, Daphne. So as we've discussed today, we've made some nice progress since our founding in April of 2024, and we're anticipating a number of key clinical milestones over the next few years. We've summarized the milestones here, and as we've touched on, we recently announced top-line data from our phase 1 trial of GlyphAgo. We do expect top-line data from our phase 2b trial of GlyphAllo in the first half of next year, 2027. We expect our current cash balance to provide runway through the readout, as well as the top-line readout of our phase 2a trial of GlyphAgo. We also expect that proceeds from an IPO would extend our runway through the top-line readout of the phase 2b trial of GlyphAgo and GAD, with some runway on the back end of this milestone, and at least through the initiation of our phase 3 program of GlyphAllo in MDD, including the relevant CMC ramp-up. As you might imagine, we expect that our spend would begin to ramp following the BUoy-1 readout, as we then be preparing for the phase 3 program of GlyphAllo, as well as the phase 2b trial of GlyphAgo. We've raised a total of $326 million since our inception, and we've maintained financial discipline as we've advanced our programs and grown the company. We ended 2025 last year with $234 million in cash equivalents, and investments. We're really proud of the progress we've made since closing the Series B in October of 2024, including initiating the phase 2b BUoy-1 trial of GlyphAllo and announcing positive top-line results from the phase 1 proof-of-concept trial of GlyphAgo, showing the potential of GlyphAgo to achieve therapeutic exposure of agomelatine while minimizing the dose impacting the liver. With that, I'll hand the call back over to Daphne.
Thanks, Lauren. And thank you all for your time today. We appreciate you joining our IPO Roadshow presentation, and we look forward to keeping you informed on our progress as we work to make an impact on these disabling and deadly conditions that affect our society.